Hello, and thank you for joining us today for the latest Technology Network webinar, How to Stain and Clear Large Samples for 3D Light Sheet Microscopy. I'm Tiffany Quinn, the Custom Content Manager for Technology Networks, and I'm going to be moderating the event today. I'm very pleased to have Dr. Afshin Khalili joining us today as your presenter, so a very warm welcome to you. Dr. Afshin Khalili is the Global Product Manager for Imaging Reagents at Miltenyi Biotech. He's been there since 2022, managing the portfolio of antibodies validated for imaging and tissue clearing reagents. With a PhD in neurobiology, he worked as a postdoc at the European Neuroscience Institute in Göttingen. He focused on using imaging, optogenetics, behavioral, and computational tools to study neuronal circuits. So without further ado, I will now hand over to Afshin.
Hello and welcome to our webinar series of A Journey into 3D Light Sheet Microscopy. My name is Afshin Khalili and I am a product manager for imaging reagents at Miltenyi Biotech. Today, in the first webinar of this series, I'm going to talk about how to stain and clear large samples for 3D light sheet microscopy. So first of all, what is 3D imaging and why it is essential? So if the question that we want to answer is heavily dependent on the 3D structure of the tissue, like for example, here, the vasculature system of the tissue, like brain, for example, then by doing a 2D classical immunofluorescence or immunohistochemistry of the slice, then we simply compromise the data, and what we get is a 2D frontal view of the section only, and we never have a clear understanding of the 3D structure of the whole sample. Talking about the workflow, I'm pretty sure that most of you know about the 2D or 2D imaging workflow or the classical immunohistochemistry. So imagine that we have a sample here, is a mouse brain. Mouse brain is opaque, light cannot pass through, and we need to do physical sectioning. Each section should be treated then individually, labeled with antibodies and placed on a microscope slide, and then finally it's being imaged individually as well. So this is good because it allows the microscopy of opaque tissue with the physical sectioning. Imaging is easy, and there are so many established standard methods in the labs. This is not good because we compromise with the sample morphology by physical sectioning. The 3D information that we get is poor. But there is a high manual workload because of all the sectioning steps and preparation and then imaging of each slide, and it is very likely that we miss a specific interesting structure in the tissue because simply this is sectioned out, or we have an image that slide actually. Yeah, so what if we do 3D imaging of large clear sample to have a holistic view of the whole structure of the tissue? What you see on the left side is 2D imaging of the mouse brain blood vessel system labeled by a CD31 antibody. And as you see, this is just an image of one slice, and the 3D imaging, the 3D information that you can extract from this picture is very, very low. Now, comparing this to a 3D imaging of the same sample using the same antibody, you can already see the difference between the two methods actually. So there is huge amount of data and information that we can extract from the 3D imaging, and in the 2D imaging we just simply miss it. The 3D imaging workflow usually consists of 3 steps. First step is immunostaining, immunolabeling with antibodies. The second step is tissue clearing, and the last step is microscopy using a light sheet microscope. So let's start with the first step, the immunostaining, and in this example using antibodies. Immunolabeling of thin sections with antibody is relatively straightforward. The antibody can easily penetrate a thin section and bind to the epitopes. Now, when it comes to the bigger structures or the whole tissue, then what comes to mind immediately is that the antibody would have a hard time to penetrate homogeneously all over the tissue. If you compare the thickness of the slice to the thickness of the whole tissue, then you may also agree with me. On top of that, the brain is opaque. The light cannot pass through, and we may need to also do tissue clearing. And tissue clearing itself, with the antibody labeling, they can interfere with each other and make things complex. So, as I said, the procedure for immunolabeling and also the clearing on top of that would be complex. The amount of time that you need for the labeling is a lot, and the background and irreproducibility of the data is always there. So all in all, it seems like rocket science. It seems like this is not possible to be established in the lab. So this is not true. At Miltenyi Biotech, we have a very good solution for you. We have the full solution, end-to-end solution for the workflow to be established in your lab. So please stay with me until the end. I will go through each step individually. So we fulfilled the first step. You beat the antibodies. I explained why we need to do the antibody labeling and also what would be the challenges. The second step is about clearing. Let me also explain what tissue clearing is. The tissue is opaque. Why is this opaque? The light cannot pass through because it has been reflected, refracted, or scattered, absorbed by different molecules inside the tissue. What we do during the tissue clearing is that we are removing some of these molecules, and also we are matching the outside environment's refractive index with inside. And that's why the light is passing through without being refracted at all. This is a very simple experiment explaining the same phenomena. This is a glass ball, and there are glass beads inside. There is air in the middle. The light is being refracted and then reached to our eyes. That's why we can see them. Now the experimenter comes and pour water inside. What is happening is actually that the refractive index of the water and glass are much more similar to each other than glass and air. That's why the light is not refracted anymore, or it's refracting less. That's why the glass beads are disappeared. This is actually a very similar effect that's also happening during the tissue clearing. Here's a biological example. So on top we have a non-cleared human ovarian tumor, and in bottom the same tumor is being cleared. That's why we can see the grid below the tissue actually. The third step of the 3D imaging workflow is imaging for which you need a light sheet microscope, or as an example, what we are offering at Miltenyi, an UltraMicroscope based system. Here I would like to give you an introduction about why and where we can use a light sheet microscopy. So there are different means of imaging for different sizes of the structures. For very small one, we may do electron microscope or confocal microscope, for example, for the tissue slices, or two-photon microscope. When it comes to bigger tissues or the whole animal, we may need micro CT or MRI or PET scan. However, you see a clear gap here. So if you want to study the cells or small structures inside a bigger tissue, for example, or a whole animal, then you may need a light sheet microscope. This was just an introduction. So if you would like to know more about light sheet microscope and how can you use that for the complex samples, and also about the light sheet microscope that we are offering at Miltenyi Biotech, please sign up for the third webinar of the series on the 13th of June. Now that I have explained all three steps, I would like to also explain and introduce you what we are offering for each step at Miltenyi Biotech. I told you that we have an end-to-end solution, and this is easy to use. So let's see what we have. The first step is immunostaining, for which we are offering validated antibodies for 3D immunofluorescence. These antibodies are validated for 3D applications. They have application in neuroscience and immuno-oncology. They are already validated, so there is no cost of validation for the labs. They are fluorochrome conjugated, and they are conjugated to the viodyes. Viodyes are the small and bright fluorophores of Miltenyi. They are directly conjugated, so this is optimum for direct staining, and you don't need to do any secondary staining, and this would save a lot of time from your side. They are recombinantly engineered, and this would minimizes the background noise and also maximizes the reproducibility of the data. The next step is tissue clearing, for which we are having Max Clearing Kit. Max Clearing Kit is a simple and non-toxic tissue clearing. So the kit consists of a permeabilization solution, an antibody staining solution, a clearing solution, and max imaging solution. So these are the reagents that you need for immunolabeling, for the immunostaining, and also for the tissue clearing, and finally for the imaging in the light sheet microscope. Here is a real example of how the tissue clearing works here. So you can see human ovarian carcinoma on top, and on bottom we see the adult mouse brain hemisphere, and in the course of 6 hours both of them are becoming clear, and you can see the sign below the tissue. Max Clearing Kit is highly efficient and robust tissue clearing. It is non-toxic organic solvent-based clearing. Max Clearing Kit is optimized for the whole mount staining, and we have also already published several validated protocols for different applications. For example, we have protocols for mouse brain, for organoids, human and mouse tumors, mouse lymph node, mouse lung, and intestine. whether you are an expert or a newbie, you may also benefit from another document that we have published on tips and tricks of tissue clearing. Thank you very much for your attention, and I would be happy to answer your questions.
Thank you so much for your presentation. I'm sure everyone will agree that that was brilliant. Brilliant pointers during it. So we have now moved to the Q&A session. I want to just remind everyone to submit a question. You can enter your question into the box on the right-hand side of your screen and click send, and we're going to answer as many as we can during the time that we have now. So the first question that I have that's come through is, how long does it take to do the whole pipeline, including staining and clearing?
Yeah, thanks for the question. Very interesting question. So our pipeline, for example, for the mouse brain hemisphere, this would take about 12 days, including the fixation, permeabilization, immunolabeling, and clearing. So all in all, about 12 days. And most of it is like about, I think, 7 to 8 days only for the labeling. And as I mentioned before, this is what we are offering is direct labeling. So you don't need to do the secondary immunolabeling or secondary antibody there. And if you want to do that, the secondary on top of that, which with our antibodies, you don't need it actually. But if you want to use like other vendors, for example, then you have to do the secondary as well. And then this doubles the amount of time you need for the labeling only.
Okay, that makes sense. So have you found labeling before clearing works better than clearing first? As we were curious as to why you chose that order for the protocol.
Yeah, thanks again. This is an excellent question. So for the labeling, you actually need to do the labeling should be done in aqueous solution. And when you do the clearing, one important step is dehydration of the sample. So when you dehydrate, then you cannot do the immunolabeling. And that's why, so what we do is first, we do the antibody labeling, and then the second is doing the clearing. That's why we chose this order.
So we've got time for one more. With 8 days of staining, does this allow for a more diluted antibody concentration, or is the elongated incubation time for antibody penetration throughout the tissue, since the protocol is optimized for whole mounting? I hope that makes sense to you.
So the thing is, we are offering the standard concentration, so recommended dilution that we have. Antibody dilution is a key point for 3D labeling, actually, because if you go slightly above the concentration, then you would label on the rim, and if you go below that, then you don't get any signal. So so we think that the concentration that we recommend, together with the number of days that we have in our standard protocols, this gives the optimum condition for the labeling.
Perfect. No, that's great. Thank you so much. So that we've actually run out of time. That's all we've got time for today. Thank you so much for all the questions, for coming in. That was brilliant. And thank you obviously so much for your presentation.